NVA237, generically known as glycopyrrolate inhalation powder, belongs to a class of drugs called long-acting muscarinic antagonists, or LAMAs. The clinical target for this molecule is the long-term maintenance treatment of airflow obstruction in patients suffering from chronic obstructive pulmonary disease, including chronic bronchitis and emphysema. Inhaled therapeutics present a unique physiological challenge. The desired clinical response must occur locally, directly inside the lung tissue, while the adverse safety risks are driven entirely by how much of the drug escapes into systemic circulation. With oral medications, a simple blood draw can predict how well a drug will work, but with inhaled drugs, systemic plasma concentration does not directly predict localized pulmonary efficacy. A high plasma concentration might only indicate a high risk of side effects, not better breathing. The FDA's Clinical Pharmacology Review of NDA 207923 documents exactly how regulators navigated this tension. To approve the drug, investigators had to map two critical vectors: establishing a strict pharmacodynamic threshold to select the lowest effective dose, and profiling the drug's complete absorption, distribution, metabolism, and excretion architecture. The final prescribing label is the result of reverse engineering the strict mathematical and physiological boundaries that separate respiratory benefit from systemic toxicity. To evaluate NVA237's efficacy, researchers relied on a primary pharmacodynamic endpoint, the change from baseline in trough FEV1, which measures the volume of air forcefully exhaled in 1 second. The clinical threshold for this metric is rigid. To prove efficacy, the drug needed to demonstrate a clinically important improvement of greater than 0.100 liters over a placebo. Finding the regimen that could achieve this required running 2 Phase 2 dose-ranging trials, labeled Study A2205 and A2208. Investigators tested a matrix of dosing options. They explored doses ranging from 12.5 up to 100 micrograms, administered either once daily or twice daily. Regulators looked for a specific data point, the lowest systemic exposure capable of clearing that 0.100 liter threshold. The data isolated the 12.5 microgram twice-daily regimen. It was the lowest dose tested that achieved an improvement of 0.139 liters, placing it comfortably above the requirement. But peaking above the threshold isn't enough. For a maintenance therapy, the bronchodilation must be sustained over the entire 12-hour dosing interval. This chart shows time course data for the 12.5 microgram dose. The solid NVA237 line maintains a sustained FEV1 improvement against placebo across 12 hours. Notice the drug's rapid onset of action. A steep initial climb occurs within the first 5 minutes post-dose. The 12.5 microgram twice-daily dose was mandated by the data. It represents the mathematical floor for localized clinical efficacy. Once local efficacy in the lungs is established, the regulatory focus shifts to mapping the drug's systemic pharmacokinetic footprint. When administered via the Concept 1 device, NVA237 has an absolute bioavailability of approximately 40%. That 40% bioavailability is split between 2 distinct pathways. The primary pathway accounts for 90% of systemic exposure, absorbed directly through lung tissue into the bloodstream, completely bypassing the liver. The secondary pathway handles the remaining 10%, inadvertently swallowed and entering the gastrointestinal tract. This GI pathway features a highly specific metabolic quirk. It undergoes immediate pre-systemic hydrolysis. As the swallowed fraction processes through the digestive system, hydrolysis breaks it down, converting it into a pharmacologically inactive metabolite known as M9. Regulators confirmed this mechanism using activated charcoal. When subjects took charcoal to block GI absorption, the M9 metabolite completely vanished from their plasma. This proved the inactive M9 originates exclusively from the swallowed portion of the drug. This graph shows plasma concentration over time for the primary lung fraction, illustrating the sheer speed active drug enters systemic circulation following inhalation. Across all demographics and doses, the drug achieves its peak plasma concentration, or Cmax, in exactly 5 minutes. This bimodal absorption effectively insulates the patient. The active drug reaches the lungs instantly, while the body neutralizes the swallowed fraction before it can trigger gastrointestinal side effects. After the absorption and distribution, the active glycopyrronium must be cleared from the systemic circulation. Between 60 and 70% of this systemic clearance is handled by a single mechanism, the renal excretion of the unchanged parent drug. Inside the kidneys, cationic active transporters, specifically OCT2 and MATE1, pump glycopyrronium molecules directly from the blood into the urine. Because the drug relies so heavily on these renal transporters, it does not depend on CYP450 liver enzymes for metabolism. Bypassing the liver enzymes gives NVA237 an incredibly low potential for drug-drug interactions, even when co-administered with other common COPD therapies like indacaterol. While variables like patient age and body weight do cause slight fluctuations in systemic exposure, the changes are too small to warrant any dose adjustments. There is only one critical failure point in this clearance architecture: severe renal impairment. This chart tracks total systemic exposure, measured as AUC, across varying levels of kidney function. Patients with mild to moderate impairment show minor changes, but for patients with severe renal impairment or end-stage renal disease, the systemic exposure more than doubles. Because the OCT2 and MATE1 transporters form a rigid clearance bottleneck, the FDA mandated specific cautionary labeling exclusively for patients whose renal pathways are severely compromised. The clinical pharmacology review of NVA237 demonstrates how raw metabolic and respiratory data are synthesized into actionable prescribing guidelines. Clinical efficacy was locked in by mathematically proving the 12.5 microgram twice-daily dose as the absolute floor required for sustained bronchodilation. Safety parameters were defined by mapping the bimodal absorption split, which protects the gastrointestinal tract, and identifying the rigid renal clearance bottleneck. By bypassing CYP450 liver enzymes entirely, the molecule remains a highly targeted and predictable therapy for COPD patients, who are often managing multiple medications. An FDA drug label is a precise biological map. It instructs physicians on exactly how to balance the necessity of localized respiratory relief against the complex architecture of systemic safety.